So tonight's focus is going to be cellular respiration, mostly aerobic or oxidative respiration. We'll look a little bit at fermentation, but again, our focus is mostly going to be that aerobic or oxidative respiration. So keeping in mind, remember, we can produce ATP through fermentation. Both of these processes, both fermentation as well as aerobic or cellular respiration, both of them are going to produce ATP. Right? I'm going to produce ATP when I do cellular respiration or when I am doing fermentation. And with fermentation, this is going to allow organisms to produce ATP if there's no oxygen available. Fermentation is going to use biomolecules in the form of pyruvate. If you'll remember, the first step in respiration is to take glucose and break it down into 2 pyruvate molecules. And so fermentation is going to allow that process of breaking down the glucose into the 2 pyruvate to release 2 ATP as well as releasing some NADH. And it's going to allow these organisms then that don't have any mitochondria, or maybe they don't even have access to oxygen. Maybe it's us and we've run out of oxygen. But they will allow the production of ATP to continue or to occur in the first place by taking that glucose and breaking it down into pyruvate. That's where the ATP will be released. Remember, this process here is going to release a net gain of 2 ATP. It's also going to release this NADH. The fermentation part is the fact that these 2 pyruvate are not then going into the mitochondria to be broken down further, but they're going to start being recycled. So there's 2 types of fermentation. We have alcohol fermentation and lactic acid fermentation. Remember, alcohol fermentation will produce ethyl alcohol and CO2. If you did the grape demonstration in your classes last year where the baggie filled with air, filled with the CO2, and it had that alcohol type smell after the fermentation was done, when we added the yeast to the bag of grapes. There's also lactic acid fermentation, which produces lactic acid. It does not produce CO2. This is what happens in animal muscle cells when we run out of oxygen. And we'll look at some summary diagrams of this as well. Fermentation, again, is not going to be your main focus. What you need to realize with fermentation is that you're still producing ATP, mainly during that glycolysis step. So it still allows organisms to produce ATP, even if they don't have a mitochondria, even if they don't have access to oxygen. A cellular or aerobic respiration is going to allow organisms to produce ATP when oxygen is available to them. This is going to use biomolecules like glucose being broken down into pyruvate, as well as 2 kinds of electron carriers, both the NADH and the FADH2. And we're going to look at how those will come into play and why those make a difference in creating so much more ATP than fermentation will. What we also need to realize about these 2 things is that all forms of life are going to have to use at least one of these processes. All forms of life have to create ATP. Plants, animals, all bacteria, protists, fungus, they all have to create ATP. And so they're going to have to do that either via fermentation or aerobic respiration. So just like photosynthesis, respiration is controlled by enzymes. It's a series of chemical reactions. Therefore, enzymes are going to control it. They're going to help make it go in a step-by-step fashion. They're going to help harness the energy from other reactions to power. It goes through, this process is a series of oxidation and reduction reactions. And I know, again, that some of you are currently in chemistry or have been out of chemistry for a long time. However, I do know those of you in chemistry, you're covering oxidation and reductions right now. Okay, remember oxidation, your oil rig, oxidation reactions, I'm losing electrons. Reduction reactions, I'm gaining electrons, right? Oil, oxidation is losing, reduction is gaining. So in these oxidation reduction reactions, what you're doing is passing these electrons on. Remember we said a few seconds ago that FADH2 and NADH were electron carriers? Well, as they pass these molecules on, just like we talked about in photosynthesis. We talked about the electron transport chain in photosynthesis that would pump hydrogens across so that it would create that hydrogen gradient, and then it would help create ATP at the end by them traveling through that ATP synthase. You have a similar series of oxidation reduction reactions, passing those electrons from molecule to molecule. So one is getting oxidized, losing its electrons. One is getting reduced by gaining its electrons. In these series of reactions, these energy transfers are going to power these electron transport chains, which are going to create ATP. So let's take a little bit of a closer look here at glycolysis. So glycolysis is the actual breakdown of the glucose, and this will occur in the cytosol. So all kinds of organisms can do glycolysis. Prokaryotes, eukaryotes, they all have cytoplasm. So this is occurring in the cytoplasm or the cytosol. We're taking the glucose, so you can see right here, we're taking the glucose, and our final end result is going to be 2 pyruvates. So again, this is all happening in the cytosol. I'm taking the glucose and I'm getting 2 pyruvates. And in that process, I get a net gain, right? It took, you can see right here, it took 2 ATP, but in the energy payoff, I got 4 ATP. So I have a net gain of 2 ATP each time I go through glycolysis. So I get a little bit of energy here. This is where the ATP come from during fermentation. Once that pyruvate is made, they don't break that pyruvate down further to make more ATP. The ATP come from these 2 that are released here during glycolysis. It also produces NADH. We mentioned a second ago that NADH is an electron carrier. So that will allow NADH. You'll notice it was you had 2 NAD plus molecules. Remember, electrons have 1 negative charge. So if I add 1 electron, that negates it, and then a hydrogen also can hold an electron. So there's where my pair of electrons go. So now I have this NADH here that is now carrying electrons for me. So this NAD plus got reduced, right? Reduced is gaining. Okay? And so there's a reduction reaction happening there. So it produces this NADH, this electron carrier, and I get 2 pyruvate molecules as an end product. If you remember, glucose is C6H12O6. Pyruvate is a 3-carbon sugar, so that's why we produce 2 pyruvate molecules. Basically, your process of glycolysis is to take the 6 carbons in glucose, rearrange them into 2 3-carbon pyruvate molecules. So if oxygen is present, because again, this is going to be part of the cellular respiration. If oxygen is present, this pyruvate here is going to move into the mitochondria. You can see I've got my cytosol out here. This is my mitochondria there. I've got a transport protein on the membrane of the mitochondria. Remember the mitochondria structure? It was a double membraned structure. So I've got this transport protein here allowing it into the mitochondria in the first place. Okay, and so that pyruvate is shuttled then into the mitochondria. And as it goes into the mitochondria, that pyruvate is going to be oxidized. Remember, oxidized is losing. So it's going to lose its electrons. You can see that happening right here. The NAD plus got reduced. Okay, and in that process of losing the electrons, you'll notice carbon is coming out. So there I've released carbon. The NAD plus got reduced. It's gaining an electron. So the pyruvate was oxidized. So now I end up with a 2-carbon molecule. Remember, pyruvate, you can see it over here, 3 carbons. Okay, CO2 is released. Okay, I get this 2-carbon molecule called acetyl-CoA. Acetyl-CoA is what's actually going to enter the Krebs cycle. So we talked, your freshman year, we talked about glycolysis, when we talked about pyruvate going in and going into the Krebs cycle, but we kind of skipped this step here. So this is a new step here, realizing that this pyruvate is going to release CO2 from it. It's going to reduce the NADH, so give some of its electrons to the NAD plus to reduce it to NADH, and it's going to become acetyl-CoA. So now that Krebs cycle, that acetyl-CoA is able to enter the Krebs cycle. You can see here's the process we just talked about right here. We're in the mitochondria now. All of this is happening in the mitochondria. Okay? The acetyl-CoA is entering now the Krebs cycle. And as the Krebs cycle turns, this is happening in the matrix of the mitochondria, which remember is located, here's our mitochondria, right? Here's the matrix here. So this is a Krebs cycle is occurring in the matrix of the mitochondria. As the Krebs cycle turns, CO2 is released. If we remember our big picture of respiration, right? It was glucose plus oxygen will give us CO2 plus water plus about 34 ATP. So here's where that CO2 is coming from as this Krebs cycle is turning. You'll also notice you've got a lot of reduction, oxidation, reduction reactions happening here. Molecules are getting, we could go through all the molecules and all the steps, all the enzymes that are in the Krebs cycle as it turns, but you don't need to know that, so we're not going to. Okay? What you need to realize is that you've got these oxidation, reduction reactions happening here. Molecules throughout the Krebs cycle are being oxidized, and these NAD pluses are being reduced, so they're gaining electrons, as well as these FADs are being reduced. So they are also gaining electrons. So the Krebs cycle is not, you're going to produce ATP, but just a little. You'll notice there's one little ATP comes off every time it turns. And remember, for every glucose molecule, the Krebs cycle will turn 2 times. Because remember, the glucose molecule got broken into 2 pyruvates. So each pyruvate gets converted into an acetyl-CoA. Each acetyl-CoA can spin the Krebs cycle. So the Krebs cycle will spin 2 times. So you get a couple of ATP, but you're getting lots of these electron carriers. And those electron carriers are really important for that electron transport chain. So when we make ATP here, we're talking about just a few. Okay? We're making the CO2 that comes out of aerobic respiration, but we're making lots and lots of these electron carriers. So these electrons are getting transferred to the electron transport chain. Okay? This is what I was talking about when I mentioned we could talk about all the different enzymes, all the different molecules. You don't need to know those. Again, what you need to realize is that you are allowing electrons, you're getting electrons into these electron carriers. You're reducing these electron carriers and allowing these electrons to get transported to the actual electron transport chain. So the electron transport chain that occurs in cellular respiration is almost exactly the same as the one that occurs in photosynthesis. So this is going to be right here. This is our inner mitochondrial membrane. So this is happening on that inner membrane. You can see where you've zoomed in here. So it's happening on the inner mitochondrial membrane here. So this darker part here would be the matrix, where all those electron carriers were being created during the Krebs cycle, right? And this would be the inner membrane space, this lighter tan here. So the electron transport chain, again, is going to act the same as it does in photosynthesis. The electron carriers will drop off their electrons at the proteins, and the proteins will then pass those electrons down the chain. So you have some proteins, these bigger, larger ones, those are going to be stable. They're not going to move anywhere, and you'll notice they have that blue arrow that's pointing up, because these are the proteins that are pumping hydrogen ions across the membrane. So as the electrons are passed to these proteins, those proteins will use that energy to pump hydrogen across. These smaller proteins, these little ones that look like little spaceships in the middle there, those smaller proteins, those can slide back and forth, and those proteins just act as transfer stations. They just help move the electrons from one protein to the other. You don't need to name all these proteins. That's part of why some of this stuff is blocked out on this diagram. So we're not trying to name all of these proteins. That's not something that you guys need to do. Okay, so we have our electron carriers, right? Our electron carrier will come over. Okay, it's going to come over and it's going to drop the electrons off, and it's going to have hydrogens available, right, for the protein to pump across. And our end result here is going to be an NADH plus molecule that can now go back and get reused. It can now go back and get more electrons. Okay, and then what we have entering a little bit later will be the FADH2s. So the FADH2s get dropped off over here. So right at this little, at the first little transfer protein. So the NADH2s get dropped off first. The NADHs get dropped off first, and then the FADH2 will jump in and drop off its electrons. And so again, no matter where the electrons came from, though, they're pumping hydrogens across at those large proteins as they're being passed down. And these electrons are being pulled down this electron transport chain because you have oxygen waiting at the end. And the oxygen that's waiting at the end, okay, is going to grab a hold. It's going to pull, it's very electronegative. It's going to pull those electrons toward it. It's attracting those electrons. Remember water, how the electrons spent more time around oxygen than they did around the hydrogen because the oxygen was more electronegative. So it's pulling those electrons towards it, which is what is making those electrons move through those proteins down this direction because they're being pulled towards that oxygen. And so that oxygen will finally grab those electrons. And when it grabs those electrons, it'll also grab a couple of hydrogen ions, and this is where the water will come out. So this is why, if there's no oxygen present, this is why the pyruvate doesn't even go into the mitochondria, because it can't run this electron transport chain. There's no reason to create the carriers if the carriers don't have anywhere to drop the electrons off. The electrons are being dropped off because the oxygen is going to pull them down. So we have to have oxygen present to do this part. So again, just like in photosynthesis though, we're creating this gradient. We're pumping all these hydrogens across into that space, into that intermembrane space. And so it's creating this electrochemical gradient. I'm going to have a chemical difference. I'm going to have more hydrogens on one side. I'm also going to have a electrical difference where I'm going to have all these positive hydrogens crammed together on one side. So they're naturally going to want to get away from each other. They're going to naturally want to spread out. And they're going to do that in the same manner that it did in photosynthesis. They're going to go through that ATP synthase so that they can produce ATP. So again, that concept of chemiosmosis. We're forming ATP because we've created, so again, this is going to be that inner membrane of the mitochondria. It could also be the thylakoid membrane of the chloroplast, right? But we've created this hydrogen gradient, this proton gradient on one side of the membrane, okay? And so the only way they can diffuse back across is through this enzyme here, which is ATP synthase. Remember the ACE part here should tell you it's an enzyme. So in synthase, right, it's going to synthesize ATP. So as those hydrogens flow through that ATP, it will actually make it spin. Okay? It will spin like a carnival ride. And so it will spin, and that spinning will help phosphorylate ADP with a phosphate, with an inorganic phosphate to create ATP. Again, this is the exact same mechanism that's happening when ATP is created during photosynthesis. The gradient is created, the hydrogens flow back through the ATP synthase, which causes the ATP synthase to spin, which will help phosphorylate the ADP into ATP. We talked about in class how this is a way that endothermic organisms are helping regulate body temperature. Okay, remember, we're producing heat during this reaction. All energy transfers aren't 100% efficient, but we can't destroy energy, right? So it can be lost, though, as heat. It can be converted into heat energy. So the inefficiency of this reaction, the not 100% part of this reaction allows heat to be released, which is what's going to allow endothermic organisms to help maintain their body temperature. That's why we talked about them needing so many more calories than an exothermic organism, because that exothermic organism isn't trying to generate the heat. It doesn't need to burn the calories. It doesn't need to create as much ATP. So here we can kind of wrap things up again. Just a reminder that oxygen is going to act as our final electron acceptor here. Those hydrogen ions are moving through that ATP synthase, both because they're attracted to the electronegative oxygen and the fact that they don't want to be there. They want to naturally spread out and get away from each other. So aerobic prokaryotes, so this is a prokaryotic organism, so it won't have a mitochondria here. Also, are you going to use oxygen for this to be their final electron acceptors? But an anaerobic prokaryote is going to use something other than oxygen to be its final electron acceptor. Okay, and so this conversion of ATP, okay, so we're making ATP, right? We're converting ADP into ATP, and then our bodies will, right here, will go back and they will reverse that, right? They will take that ATP and convert it back into ADP, an inorganic phosphate, and that's where we get our energy for cellular work. Okay, so we break down molecules, we do catabolism or digestion, break down molecules to help create ATP, and then use that ATP for our cells to actually be able to do work. So big picture reminder here. Okay, first of all, this is all part of our mitochondria. Remember, this is the mitochondrial space, the light tan. The darker brown is the matrix. Right, so let's say off on the white, on the edge, let's say this is our outer membrane. So the white then would be our cytoplasm. So we had glycolysis happening out here in the cytoplasm. And then if oxygen is present, right, that pyruvate got pumped into the mitochondria. And that pyruvate is going to get converted into acetyl-CoA. Remember, that pyruvate is going to get oxidized, and it's going to reduce some NADH. So I get some electron carriers there, right? The Krebs cycle makes more electron carriers, both NADH and FADH2. Remember, I also got some NADH during glycolysis that will head to the electron transport chain. Okay, so NADH is made more often than the FADH2. We get a little bit of ATP from that Krebs cycle. Okay, we also get that CO2. Okay, and then the electron transport chain is occurring. So here's my electron transport chain. Again, this is why, remember the structure function, and this membrane is all convoluted and bent and twisted so that it has more surface area. More surface area allows for more of these electron transport chains to be happening. So that electron transport chain is pumping hydrogens across into the inner membrane space of the mitochondria, from the matrix into the inner membrane space. Okay, my carriers are dropping off their electrons. Their electrons are getting pulled down the electron transport chain by this electronegative oxygen, which is going to pair up with some of these hydrogen ions to become water. So we're going to release water here. We needed oxygen to act as the electron acceptor, and then it's going to release water. This is my ATP synthase over here at the end of my chain. So those hydrogen ions will diffuse back through the ATP synthase. That will turn it on. Right? We'll take the ADP in an inorganic phosphate, and we will convert that into ATP. So don't lose sight of your big picture. Right? Hopefully at some point you've drawn some of this, but again, don't lose sight of your big picture. Right? For oxidative or aerobic respiration, I require glucose. I require oxygen at that electron transport chain to be my final electron acceptor. I'm going to produce water when those electrons are bound to that oxygen. I'm going to produce CO2 when I do some of my carbon rearranging. And I'm going to produce a whole bunch of ATP. If you add them all up, about 36 to 38. And that varies because of the electron transfers, the electron carriers that are dropped off, okay, and as the electrons pull through. So we'll do some more comparisons with this. We'll look at it compared to photosynthesis. We'll do some summary diagrams and stuff in class.